And the questions will be in the comment box, right?
Yes, it will be in the comments box. And at the end of the day, I will highlight them and I'll read out as well.
Okay.
Wow, that's amazing. 17 people already logged in.
Oh, that's cool.
I think it's 8:30, so we can start with that. Just 2 minutes. All right, good morning, good afternoon, and good evening to everyone from wherever you're listening to, and a warm welcome to the Biologically Speaking. Here we discuss the progress and prospects of biological research through regular seminars and blog posts. It's an academic research interest group that has been brought to you by a group of PhD and postdoctoral fellows from nearly all over the world. And today is a special day, and greetings of the National Science Day to you all. The day is to commemorate the achievements of science in the society, and we are delighted to have Dr. Deepak from ICMR-NIRRH. Deepak does not need an introduction, but just for the sake of the platform and for the whole webinar, Dr. Deepak earned his PhD in human genetics from University of Mumbai in 2003 and joined the ICMR National Institute for Research in Reproductive Health in Mumbai. His lab is interested in understanding the molecular basis of embryo endometrial crosstalk and implantation and early placentation, and how these processes go awry, leading to infertility, recurrent implantation failure, endometriosis, and preterm births. And also he is addressing the genetic basis of disorders of sex development and male infertility. Dr. Modi has received numerous prestigious awards, including the Indian National Academy of Science Young Scientist Medal in 2006, the Platinum Jubilee Young Scientist Award from the National Academy of Science India in 2007, the Innovative Young Biotechnologist Award for the year 2007, the ICMR Tilak Venkoba Rao Award in 2010, and recently being elected as the Fellow of the National Academy of Sciences India in 2020. And I think this is his first talk after the official induction yesterday he had from the National Academy of Sciences in India. So we are delighted to have him. And his lab played a major role during the pandemic on generating the COVID registry for the pregnant women and how COVID-19 can affect maternal immunity as well as pregnant women. So we're all delighted to hear his talk today. Is on placenta, pregnancy, and COVID-19, and we're all excited to hear from you, Dr. Modi. So the screen is all yours. We are very much looking forward to it. Thank you. Just a quick reminder to all the participants who are joining through YouTube, please post your questions for Deepak Modi during the talk using the chat box in the YouTube, and we will try to moderate them and cover most of the questions depending on the time and get it moderated at the end of the today's session. Hope you will all have, hope you'll all enjoy today's session. So looking forward to it. Dr. Modi, over to you.
Thank you. Thank you so much, Shaunak. And it's a pleasure and it's an honor to speak on the National Science Day in remembrance of the great Raman. And thank you, everyone, for joining me on a beautiful Sunday, spending time. And again, like as Shaunak said, good evening, good afternoon, and good morning from whichever part of the world you are in. It's an immense pleasure. And thanks, Biologically Speaking team, for having me on board. And this is some work that we have done in the last 1 year on, as Shaunak said, COVID-19 and pregnancy. And in the last next 40 minutes, I'll try to talk about some of the things that we have achieved in this area. I hope my slides are visible, Shaunak.
Yes, they are perfectly fine.
Okay. So my lab, as Shaunak said, is interested in looking at basically embryo implantation and placentation, and how things go wrong in these processes leading to pregnancy-related disorders or infertility. And more recently, we have got interested in COVID-19 and pregnancy. And the other aspect that we are looking at is disorders of sex development and infertility. To do this work, I have a wonderful team of students and postdocs who really work hard and make the lab very proud. And we have a number of publications, presentations, and I'm sure you've seen some of them in the last couple of years. And today's talk that I'm going to give is done by almost every student of mine during the pandemic, and they've all contributed very nicely. Of them, special thanks to Anshul, who really came in the midst of the lockdown and even worked hard to get some of the data that I'll be presenting today. So without much ado, when we talk about pregnancy is quite a different status, because here the maternal immune system is compromised because it has to accommodate a fetus which is not, which is semi-allogenic, and therefore the mothers are more likely to be infected and may have adverse pregnancy outcomes. And along with adverse pregnancy outcomes, there is also a chance in several instances that the virus from the mother can get into the baby, which we call it as vertical transmission. For example, the Zika virus infection during pregnancy, the virus can get into the baby and then cause brain defects. For HCV, the HCV virus can have an intrapartum infection, and it could lead to miscarriages or fetal losses. So traditionally, viral infections during pregnancy are known to be detrimental, and there could be a congenital infection, that is infection from the mother to the baby during pregnancy, which could lead to growth retardation or fetal losses. There could be perinatal infection, that is during late trimester or at near birth, and that could lead to fetal meningitis, septicemia, pneumonia, or preterm labor. Or there could be an infection just around at the time of birth, and which could also have adverse pregnancy outcomes. Now, when it comes to SARS-CoV-2, initial reports suggested that this is a disorders of old and young people are not really infected much, and therefore not much attention was paid to pregnant women. But if you look at now the current data, we realize that almost 60% of the total SARS-CoV-2 infections in India, for example, or as well in the world, is in the reproductive age group, and therefore it is not surprising that pregnant women also do get infected by SARS-CoV-2. So when this pandemic began, we did not have much idea about anything. We were not even sure whether pregnant women would get infected with SARS-CoV-2. So we asked a very simple question, that can SARS-CoV-2 have any adverse effect on pregnancy? So it's a very basic question we started our work with. And we wanted to see whether the SARS-CoV-2 virus can infect the developing embryo at very early gestation ages when the pregnancy starts, or can it pass on from, and during pregnancy if a woman gets infected, can the virus pass on from the mother to the baby? Could there be transplacental infection? Or if there's a late trimester infection, could there be a transplacental infection, or could there be any adverse outcomes on the baby? So the first question that we asked was, are human embryos permissive to SARS-CoV-2 infection? So now before I go ahead, let me give a quick overview of how SARS-CoV-2 enters inside the cells. So this is a virus which has a receptor-mediated endocytosis process where the virus binds to its receptors on the cell surface, so it kind of catches the virus. There is another enzyme which you require to process the viral protein, and then the virus gets inside the cells, it releases its contents, it replicates there, and then it is thrown back out. So this is what we call it as a viral life cycle. This is true for almost all coronaviruses, including SARS-CoV-2. Now to do this process, now the virus just cannot get inside the cells because the cells have a nice plasma membrane barrier, but it requires a protein called as ACE2. It requires an enzyme called as TMPRSS2, which is required to cleave the S proteins. And these are the two main canonical proteins of the cells which are used by SARS-CoV-2 to enter in. Over and above that, there are some non-canonical receptors like CD147 or Bayesigen. The non-canonical S protein cleavage proteins are cathepsins and furins. And so we started looking whether these proteins or RNA for these proteins are expressed in the early developing embryos. The idea was to see if these proteins are present, then it is likely that the SARS-CoV-2 can enter in these cells. So to do this, we employed a very simple strategy. The good part is of data sharing, where people do a lot of experiments and put this data on the World Wide Web. And what my students thought was that there is single cell RNA-seq data of developing human embryos, which is very nice. So in this, what it's done is human embryos which are donated for research, every single cell of the human embryo is separated, sequenced, and now we have RNA information of every cell of the developing human embryos. What my students did, by this time lockdown was imposed and they were all sitting in their homes, but what they did was they downloaded these data sets of human embryos which had single cell RNA-seq data sets of human embryos, and they started searching for cells which were expressing these SARS-CoV-2 receptors. And here was the data, and this was very, very surprising for us. Now, the human embryos have several stages. This is zygote stage, which is just after fertilization. There is 4-cell stage, 8-cell stage, compact morula, and the blastocyst. And in each of these cell types, we started looking for presence of ACE2 protein in these cells. And interestingly, what we found was that even at the time of fertilization or 4-cell stage, these embryos express ACE2, which is a receptor required for SARS-CoV-2 infection. And then the levels go down, but again, as the embryo starts growing, the number of ACE2 positive cells increase significantly. TMPRSS2, as I told you, is required for S protein cleavage. And we see that at the early developing stages, the TMPRSS2 is absent, but as the embryo starts growing and gets up to the blastocyst stage, the number of ACE2 positive cells increase significantly. The alternate receptor Bayesigen is expressed by almost every cell type of the developing embryo. Now remember, what I've told you right now are these cells which are inside the embryo. The placenta which is formed is because of this trophectodermal layer, what I'm showing you in green color here, and this placenta, and these are the future placental cells. So the question that we asked was that, are these trophectodermal cells also expressing these receptors? And to our surprise, more than the embryo proper, the trophectodermal cells of the developing blastocyst, that is from day 5, day 6, and day 7, remember this is all human data, abundantly express ACE2 mRNA. The TMPRSS2 is also very nicely there, and Bayesigen is also there. This was convincing evidence to tell us that the developing human embryos are permissive to SARS-CoV-2 infection. Now remember, it is not just sufficient to have the receptors present, but the receptors also have to let the virus go inside, let it replicate, and then multiply. So we asked whether and for this entire process, it reuses a lot of other cellular proteins, the host proteins. So we asked whether the host proteins which were required for viral endocytosis and exocytosis are present in the developing human embryos. And here is the data. You see here, these are early developing embryos, and there is very little expression of the endocytosis proteins. But by the blastocyst stage, almost every protein which is required for endocytosis of viruses are abundantly expressed, mainly in the trophectodermal cells. By the time we were doing this work, there were 30 proteins identified which were required for SARS-CoV-2 replication, and about 333 proteins required for interaction with SARS-CoV-2 proteins so that the virus can either replicate or use the host machinery for its exocytosis. And we asked whether these mRNA or proteins are also present in the developing embryos. And look at this data. I showed you the data of viral endocytosis, but even the viral replication genes are present in the developing human embryos, mainly at the blastocyst stage. And even the SARS interactome genes, that is, these are proteins or human proteins which are interacting with SARS virus proteins, and they do a whole lot of things in that process, affect cellular metabolism in the cells, and all these proteins are abundantly expressed in the developing embryos, mainly at the blastocyst stage. So based on our data, we suspect that either the early zygote or specifically the blastocyst are permissive to SARS-CoV-2 infection. And my students also worked out and said that it's not just SARS-CoV-2, but these stages of the embryos are also possibly susceptible to MERS as well as the other coronaviruses also. So now I'll switch gears. Up to this point, we said that even as early in pregnancy, when the mother has not even realized that she is pregnant, and if she gets infected by SARS-CoV-2, and somehow if the embryos get SARS-CoV-2 experience SARS-CoV-2 in its vicinity, then the embryos can get infected, and there is now data to show that such an infection can lead to early embryonic death, which could lead to infertility-like conditions or miscarriages. This data is very crucial for IVF clinics. Remember that during the process of IVF, where the mother's eggs and father's sperm are taken and the embryos are developed in the lab dish, during this course, if somebody infected is in the laboratory, remember many of these SARS-CoV-2 infections are asymptomatic, and if there is somebody infected in the laboratory who doesn't know is a carrier of the infection, might spread the infection in the lab, and even the IVF embryos can get impacted. So in the IVF clinics, this data becomes very, very important. But now let's get to the main topic, whether the early developing embryos might be susceptible to SARS-CoV-2 infection, but whether the embryonic stages during pregnancy, now let's say that the mother is pregnant and she gets infected with SARS-CoV-2, what could be the outcomes? So this is a very interesting thing. Now, pregnancy is a very interesting situation because it's not very easy to pass on things to the baby, and baby cannot send things inside. Now, when the mother is pregnant, the baby develops an organ called as the placenta. Now, this placenta, it acts as a very nice barrier. It does not allow things to easily pass on from the mother to the baby and baby to the mother. And therefore, it acts as a nice protective shield around, which protects the baby from multiple infections. The question that we asked was that, can placenta be permissive to SARS-CoV-2 infection? If the placenta is permissive, then it's likely that there could be mother-to-child transmission of SARS-CoV-2. But if the placenta is not permissive, then the likelihood that the baby gets infected is very, very low. So the next question that we asked was, is placenta permissive to SARS-CoV-2 infection? Now, before I go ahead with the results, I'll give a very, very quick lesson on the placental structure. My apologies to those who already know the placenta, but it's nice to orient ourselves in this direction. So here is the placenta which is developed during pregnancy by the baby from, remember I told you, the trophectodermal cells which make the placenta. These placenta form placental villi. The fetal cord will have the fetal blood vessels which go inside the placenta, and these villi now float inside the mother's womb. It is not the amniotic fluid, but here is where the mother is sending out blood. So this is the mother's uterus, where the mother is sending out blood. And remember that there is no blood exchange, but all the nice nutrients from the blood are absorbed and given to the baby through these placental villi. So these placental villi, you can see here, they are anchored onto the mother, and here is a mother's blood coming, and it is all floating around. So all the placental villi are bathing in maternal blood. And there are two kinds of villi. You have the anchoring villi and you have the floating villi. The anchoring villi are the cells which are going to invade into the mother's uterus further, the endometrium, and they're going to line the spiral arteries for spiral artery remodeling. And this process is very crucial, and if this process gets affected during pregnancy, there could be pregnancy losses or preeclampsia-like conditions, that is hypertension during pregnancy. And there is another set of villi, which we call it as free-floating villi, whose job is not to do this, but whose job is slightly different, whose job is to absorb the nutrients from the mother, filter them out. So this placenta is like a nice filter of the nutrients and give it to the baby. It also gets in waste products from the baby and passes back to the mother, so mother can remove them from the circulation. So the question that we asked was that, are these villi permissive to SARS-CoV-2 infection? Now, just one round of orientation, that we looked at the free-floating villi and the cells of the anchoring villi, which could be potentially leading to SARS, which could be potentially exposed to SARS-CoV-2. Remember, I told you that there is maternal blood everywhere around, and if there is virus in the blood, these cells can experience the virus, but they need not get infected if there is no ACE2 there. So we asked whether the cells of the placental villi has ACE2 receptors. Now there are multiple kinds of cells here. In the villi, the outermost layer is called as a syncytotrophoblast. Inside it are the cytotrophoblast, and here is the mesenchyme stroma, and then there are fetal blood vessels. So you realize that for the fetus to get infected, it has to pass through these multiple layers. It cannot just pass through because it's a very nice filter, very tightly bound, not anything and everything can go very easily. Now, if it has to pass, the virus has to pass on through these structures, it has to first enter in these cells. And as I told you, for entry, you require the ACE2 receptors. So parallelly, while we're doing the embryo work, my other set of students started looking at the placenta data. The good part was that by last year, by 2018, 2019, a number of placenta data sets were available on the World Wide Web. And we started downloading the data, where here we took a data from one of the papers published in 2018 called as Lee et al., where there was single-cell RNA-seq data of syncytotrophoblast, cytotrophoblast, villus stromal cells, and extravillus trophoblast, independently isolated, and all of these were then sequenced by single-cell RNA-seq technology. So our job was to take this data and start looking for the presence of these receptors in every single cell. And here is the data. Now, as I told you, extravillus trophoblast, syncytotrophoblast, syncytotrophoblast, and STRs. And here you see that not all the cells of the placenta were expressing ACE2, but a small subset of syncytotrophoblast and syncytotrophoblast were happily expressing ACE2, and there was also TMPRSS2 there. The basogen, the alternate receptors, and cathepsin L, the alternate S protein cleavage enzymes, were very nicely expressed in these cells. Now, remember, it is important that ACE2 and TMPRSS2 should not be separately expressed, but they should be expressed in the same cells. So we started looking whether how many cells of the placenta co-transcribe or have both ACE2 and TMPRSS2 on the same cell type. And to our surprise, we found that there were a proportion of syncytotrophoblast cells and the cytotrophoblast cells which were abundantly expressing ACE2 as well as TMPRSS2. And here is the immunofluorescence. So we looked at the protein data. So this blue color are the nuclei and the red color is the ACE2 protein. And we can see that the cells, some cells of this placental villi, especially the syncytotrophoblast layer, which is outside, and the cytotrophoblast layer, which is inside, had ACE2 protein. So you see, it's not all the cells which are expressing, but a small subset of cells are abundantly expressing ACE2. And once the ACE2 is there, if the virus comes, the placenta is likely to get infected. My students also looked at MERS receptors and COV229E receptors, and they found that these are also expressed in the placenta, indicating that the placenta is not just susceptible to SARS-CoV-2, but it is also susceptible to the other coronaviruses. And interestingly, the ACE2, Basigen, TMPRSS2 were not just expressed in the human placenta, but they were expressed in placenta of multiple species, including the armadillo, elephant, opossum, mouse, indicating that the expression of SARS-CoV-2 in the placenta is not just unique to humans, but it is there in placenta of every species. Now, when it's there in placenta of every species, it is likely that it is going to be functional and it is going to do some jobs. So overall, our results told us that in the first trimester at least, so remember this pregnancy is divided into first trimester, second trimester, third trimester. We had data for the first trimester, and in the first trimester at least, the extravillous trophoblasts which are going to invade into the placenta and the syncytial trophoblasts which are going to see the mother's blood are highly expressing SARS-CoV-2 and ACE2 and TMPRSS2. So this work was done with the help of Dr. Mohit Jolly at ISC Bangalore, and with the help of Priyanka in his lab, we characterized these cells further. We wanted to know what are these syncytial trophoblast cells doing. If they get infected, what could go wrong in this placental cells? And our data showed that this syncytial trophoblast cells, which are ACE2 and TMPRSS2 positive, had a very, very unique gene signature. And these cells had an important role in interferon signaling pathway. They had an important role in mitochondrial permeability. They had a role in maintaining adhesion junctions, and they also had a role in proliferation. And eventually we found that these cells are highly differentiated syncytial trophoblast cells. They're terminally differentiated. They express all the molecules which are required for glucose exchange, and they also had abundant expression of the entry molecules which are required for SARS-CoV-2, the viral replication machinery which is required for multiplication of the virus, and even the SARS-CoV-2 interacting proteins were very nicely expressed in these cells. That goes to say that the syncytial trophoblasts, which are immediately going to experience the virus, not only have the receptors, but also as a necessary machinery which will allow viral replication. And then we looked at the extravillous trophoblast also. These are the cells of the villus which are going to invade into the mother and model the spiral arteries. These cells also had a very unique gene signature, and we found that this extravillous trophoblast, which are positive for ACE2 and TMPRSS2, abundantly express the ESCRT genes, that means the viral endocytosis genes. They had a lot of genes which were required for viral replication as well as SARS-CoV-2 interaction. And more interestingly, I'm sorry for the small font here, but more interestingly, we found that these genes which are expressed, these ACE2 and TMPRSS2 positive cells, EVTs which are expressing, have very nice expression of the genes which are required for endovascular remodeling. That means if the placenta gets infected in the first trimester, it is going to infect cells which is going to line the maternal arteries, and this process can get difficult during pregnancy, and this can have long-term adverse outcomes during pregnancy. So to summarize, we show that the villus cytotrophoblast, syncytial trophoblast, proliferative EVTs, invasive extravillous trophoblast, and endovascular trophoblast abundantly express SARS-CoV-2, and of them the highest expression is seen in syncytial trophoblast and the extravillous trophoblast layer. So now this set the stage here. The stage, so we published this data in the Frontiers in Cell and Developmental Biology mid last year, sometimes in August. This paper is very well received, already cited a bunch of times. And this paper said that the placenta is most likely to get infected by SARS-CoV-2 because it has the necessary receptors. But now the question arises that, okay, fine, the receptors may be there, but the placenta, does it ever get infected? Can the placenta even get to see the virus? because if the mother has to transmit the virus to the baby, it has to go through the placenta. The placenta is necessary machinery, but just the presence of machinery is not sufficient. We needed to document whether the placenta can also get infected by the virus. Now, while we were doing these studies, we came across a very interesting case report. So it was one afternoon in, I think, June or July, where we got a phone call from one of my colleague, actually, in fact, Dr. Rahul Kachwe got a phone call from ESIS Hospital in Mumbai, from Dr. Prajakta Shende. She informed us that there is a case, very interesting case, where a pregnant woman was infected with SARS-CoV-2 when she was 8 weeks pregnant. So very early in pregnancy, she was infected with SARS-CoV-2. The pregnancy, she was asymptomatic. She did not have any symptoms. So she was home quarantined. She did not receive any medications, and she spontaneously recovered, like how most of the patients recover. But when she came back to the clinic at 13 weeks for a routine ultrasound, for a routine antenatal care, she was advised to do the normal ultrasound, which normally all women are advised to. And what she found was that there was intrauterine fetal demise. The baby was dead, and there was hydrops fetalis, which means that there was accumulation of fluid around the baby. And you can see here I marked it with arrows. There is a lot of fluid accumulation around the baby. There was no cardiac activity indicative of fetal demise. Now, this is an interesting case for us because the mother was infected when she was 8 weeks pregnant. Almost 5 weeks have passed after that, and now there is a manifestation of fetal death. And we were wondering what could be the reason. So the question that we asked was that, is the virus there in the placenta? Because by this time we had enough data to show that the placenta can get infected with SARS-CoV-2. So what we did was we told the collaborators that please send us a placental tissue, collect some amniotic fluid, and then please send it. We will test it for us. And what we found was that on the day of when she was detected positive for SARS-CoV-2, she had a very moderate viral load. It was not that she was heavily infected and symptomatic. She was moderately infected. She was totally asymptomatic. And after 5 weeks, when she underwent the abortion and the fetal death was there was no virus in her throat swab. However, when we looked at the placental tissue, we found that the virus was present there. That means the mother herself had cleared the virus from the blood, but we could detect the E gene and the N gene of virus in the placental RNA as well as the amniotic fluid. This was unprecedented. Now, this is the first case that we had come across where the first trimester infection, totally asymptomatic. The mother had nothing happening to the mother, but her baby, which was dead by this time, the placenta had harbored the virus already there. This was kind of corroborating what we had seen in our single cell RNA-seq data. But then we asked that this is not sufficient. I mean, just presence of RNA in the fluid, it could be just coming from the fluid somewhere, and the placental cells may not have actually got infected. So what Anshul did was he performed immunofluorescence for SARS-CoV-2. Now, SARS-CoV-2 has the S1 protein and the S2 protein, and we took the sections of this placenta, we stained them by immunofluorescence for SARS-CoV-2 proteins, S1 and S2, and ladies and gentlemen, here is the data for you. You can see this red color virus all in the placental villi. Remember, I had shown you the data. There is ACE2 positivity in these cells, and the same cells there was positivity of S protein, both S1 and S2. This shows that the placenta is infected with SARS-CoV-2. Actually, the virus is getting inside. And here you can see another data. This is two-color immunofluorescence localization, where we looked at another viral protein called as non-structural protein NS, nucleocapsid protein, and the green staining is for ACE2, which is the receptor, and look at the higher magnification. These are the syncytotrophoblasts, these are the cytotrophoblasts. They all are positive for ACE2. That means they're having the receptors, and the same cells are also positive for SARS-CoV-2, indicating that the virus actually interacted with this case ACE2 and got inside, and it started replicating there. The virus was replicating there because we already knew by this time that the viral replication machinery is present in the placenta. So this was the first case report which we published in Human Reproduction, and where we could show that it was not just the placenta which was getting infected, but in the first trimester, the placenta was getting infected and the virus went to the amniotic fluid, and the baby also got infected with the virus, indicating that there was a clear evidence of mother-to-child transmission of SARS-CoV-2. Uh, uh, so we further characterize these cells of the trophoblast of the first trimester. We wanted to see whether how capable are these of mounting an immune response. Remember, the mother usually has to mount an immune response to the virus, and the placenta also, if it gets infected, it may mount an immune response. I told you that the cytotrophoblast and the extravillous trophoblast are capable of SARS-CoV-2 infection. And we found that these cell types, especially the extravillous trophoblast of the first trimester placenta, were enriched for genes in the toll-like receptor signaling pathway, apoptosis pathway, the JAK-STAT signaling pathway. And these pathways are involved in cell death as well as immunomodulation. And when we looked at the histopathology of the first trimester placenta, which was infected with SARS-CoV-2, we found very interesting findings. In some of the areas, we found that there was complete lysis of the cytotrophoblast layer. That means the virus is getting inside the cells, replicating there, and there was apoptosis of this. Of course, we have not looked at apoptotic genes here, but we could see that the viral infection occurred in the cytotrophoblast layer, and there was lysis of this. Over and above that, we found that there was evacuation of the placental villi, and we could see there was a lot of fibrin deposition around the placental villi, and there was a lot of fibrin deposition even in the maternal decidua. And look at the decidual cells. There was abundant, there were many blood vessels, and these blood vessels were now kind of getting engorged with blood, and around them also there was a lot of fibrin deposition, indicating that there was hemolysis there. There was maternal vascular malperfusion happening in the first trimester placenta. And you can see here there was infiltration of lot of neutrophils and lymphocytes. The portion of the decidua, you should never see the cells in the normal pregnancy. Normal pregnancy in the decidua, these cells don't infiltrate, but in the first trimester decidua, where there was infection of SARS-CoV-2, there was infiltration of the immune cells as well. So what we show here is that if the mother gets infected with SARS-CoV-2, remember this is a respiratory virus, and the respiratory virus cannot easily get to the placenta, but in a subset of women, it is possible that it will show a systemic viremia. That means it will bypass from, it will get into the lungs, from lungs it will get shedded into the maternal circulation, and once it is into the maternal circulation, through the maternal spiral arteries, it can reach the placental villi. On the placental villi, I have shown you data to show that there are receptors, the ACE2, TMPRSS2, the alternate receptors are there, so the virus can bind onto these cells and enter them. These cells have necessary machinery for viral replication, and once a viral replicates, that the placenta gets heavily infected, and then it can go into the fetal capillaries, leading to mother-to-child transmission. So this is the first case in the first trimester we reported that there is a distinct mother-to-child transmission of the SARS-CoV-2, where the mother is infected, the virus gets into the circulation, and then infects the placenta, leading to fetal death. Now the question arises here, so there were several burning questions which were getting posed to us during this period when we were working on this, that what is the actual incidence of vertical transmission of SARS-CoV-2? Is it necessary that all the mothers who get infected pass on this virus to the baby, or there's only a small subset of mothers in whom there could be viremia and the placenta getting infected? Now, if the mother is getting infected, are there any health risks posed to the mother because of the SARS-CoV-2 infection? If the mother is getting infected, if there are no health risks, can it have any effect on the pregnancy outcome? For example, in this one case report, we had an abortion. But what are the other pregnancy outcomes that could happen in SARS-CoV-2 infection? And if the mother gets SARS-CoV-2 infection and if she passes on to the baby, what is happening to this baby? Because remember, this is just one case report, and there was very little information available by that point of time. People were saying that, oh, virus doesn't seem to be doing much to the pregnancy, but it was not turning out to be the case, because now evidences were mounting suggesting that placenta is possibly getting infected with SARS-CoV-2, and there is a likelihood of mother-to-child transmission. So to address that question, we carried out systematic review. So we looked at the world data first and tried and see that are there any health risks posed by SARS-CoV-2 infection, posed by SARS-CoV-2 infection during pregnancy. And we found very interesting results. I'm not presenting all of them, but this is one snapshot where we classified this world data into women coming from low to middle income countries and women coming from high income countries. Now, women coming from low to middle income countries are anyways at a higher risk of developing pregnancy complications. But we found that as compared to women residing in high-income countries, women residing in low to middle-income countries have a higher risk of abortion if they get SARS-CoV-2 infection. There is a higher risk of fetal distress, and there is a higher risk of placental disorders and hypothyroidism, and of course, anemia. This was very surprising because we thought that there could be higher risk of preterm births and PROM, but that was not the case. It was higher in the Western countries, but in the populations like India, mainly the LMICs, these women were higher risk of getting an abortion. So this posed a very important question to us, and by that time the Maharashtra government also started getting, because Maharashtra was a hotspot of virus, hotspot of SARS-CoV-2 infection, and there were a lot of public health concerns, and my colleague Dr. Rahul Kajbiye decided with the help of our director, Dr. Smita Mahale, that we will start the Preg-COVID registry. So this is a national registry for pregnant women in India, where the aim was to document pregnancy outcomes of Indian women with COVID-19. The idea was, just because these women would be likely to getting into the hospital, so from the hospitals, if we know that what are the pregnancy outcomes, we can develop a database and know what are the health outcomes of SARS-CoV-2 infection during pregnancy. It was a lot of task, because then we had to identify which hospitals are going to give us this data, how we are going to implement this. So we got in touch with MEDD, that is Maharashtra Drug and Educational Department, under which there are a lot of medical colleges of the government of Maharashtra, which are spanned throughout Maharashtra. So with the help of MEDD, we were able to contact these colleges and we set up this registry. So these are now totally, as of today, we have 19 medical colleges all spread all across Maharashtra, who is in real time giving us data of SARS-CoV-2 infected women who are coming to these hospitals. We are recording what are the health, what are their SARS-CoV-2 infection status. We are recording what are the symptomatic presentations, what are their pregnancy outcomes, and what is happening to these babies. This registry we had started sometimes in May. It has collected retrospective data from March, and prospectively it's going to collect till the end of next year. The idea is to document what are the pregnancy outcomes in these women with SARS-CoV-2 infection. So as soon as we set up the registry and we started collecting data from these different centers, we found a very, very interesting observation. This paper, which we published in mid 2020, with Dr. Rakesh Wagmare, who was very, very instrumental in setting up this registry, and we express our sincere gratitude to him. What we found from this data was, by that time we had data of about 1,040 pregnant women. There were 141 which were infected with SARS-CoV-2 by that point of time, indicating that the risk of getting infected during pregnancy is roughly 12%. So 12% of pregnant women are likely to get infected with SARS-CoV-2. But when we look at the symptomatic data, whether these women were symptomatic or asymptomatic, we found that almost 88.5% pregnant women were asymptomatic and only 11.5% were symptomatic, indicating that a large number of pregnant women could get infected with SARS-CoV-2, but they would remain asymptomatic throughout. And this is a problem because these women would get into the hospitals and then they would be attended by the doctors and nurses for the delivery. And if they are infected with SARS-CoV-2 and they don't know, they are going to spread this infection in the community. So based on that, with our results in hand and with a lot of other data, ICMR by that time made a mandatory policy that universal testing for SARS-CoV-2 should be done in pregnant women in India. Every single center in this country who is delivering women for SARS-CoV-2 is now screening these women for SARS-CoV-2 infection, so that they know that there is a pregnant woman who is positive for SARS-CoV-2, and she should be separated in an isolation ward, and her labor requirements are very different as compared to non-infected women who would be taken in a separate ward and a separate labor room, so that there is no chance of cross-infection. So this data which we got from the registry had a very important policy implication. And this is the first time that we showed in India that the infectivity rate in pregnant women was about 12% and the symptomatic rate was about 10%. By this point of time, this was very early data, of course. By this point of time, till December 2020, in our registry, about 24,650 women have already been screened. And from this, roughly 2,500 women have been case positive, indicating that the case positivity rate is about 10.2%. This is very satisfying to know that although pregnancy and SARS-CoV-2 might have some problem, but at least this data tells us that pregnant women are not more likely to get infected with SARS-CoV-2 infection as compared to the normal counterparts. The average Maharashtra data is was 13.2% by the end of December. So this is fairly close to it, although slightly lower. But remember, this is a cumulative data of both males and females. Females are less likely to get infected with SARS-CoV-2 in general, but at least it is gratifying to know that pregnant women are not more likely to get infected with SARS-CoV-2. Having said that, we found that the maternal death rate in our registry, this is, remember, just a snapshot. I don't have too much of details here because this data was just accumulated up to 31st December. We are still analyzing a lot of data here, but we had a maternal death of 1.1%. This is worrisome. So women who get infected with SARS-CoV-2, about 1% of them have a risk of death. Now, 1%, although sounds a small number, remember, it's not a small number, just like that. It's simply because maternal mortality rate in Maharashtra, as per Niti Aayog, which published its data in 2015 and 16, is 0.061%. That means that over and above pregnancy, so if a pregnant woman is there's only less than 0.06% chance that a pregnant woman would die, because a woman would die because of pregnancy. But if she is getting infected with SARS-CoV-2, there is a huge jump in the death rate. So if the mother gets infected with SARS-CoV-2 as compared to their uninfected counterparts, the likelihood of death is very high. Even if we look at the case fatality rate in Maharashtra of young individuals, that is between 21 to 41 years of age, is 0.6%, whereas the maternal death rate is 1.1%. So there is at least a 0.4% increase. That means there is a doubling of death rate in pregnancy. So pregnancy and SARS-CoV-2, although the women may not be very more likely to get infected with SARS-CoV-2, but if they get infected with SARS-CoV-2, their chance of death is much higher. We found there was abortion rate or stillbirth rate or loss of pregnancy rate is about 3.3%. The background stillbirth rate in Maharashtra is less than 1%. So the chance of abortion because of SARS-CoV-2 infection is about 3 times more than that of a normal pregnancy. This is unprecedented. This is the first time ever we are showing data where we show that pregnant women, although not more likely to get infected with SARS-CoV-2, if they get infected with SARS-CoV-2, there are more chances of adverse pregnancy outcomes and more chances of severe outcomes like abortion, fetal loss, or maternal death. I'm not showing you much of the data there because it's still under analysis. We have published some case reports or some interesting observations. Here the question that some of the doctors posed to us was that because during pregnancy there could be twin pregnancies as well as singleton pregnancies. Are the twin pregnancies more likely to have any adverse outcomes? Because remember, I told you that the placenta can get infected, and because of the placental infection, there could be some adverse outcomes. Now, in case of twin pregnancy as compared to the singleton counterpart, so remember these are both infected. When some women are carrying only about 859 women, we had data of them was carrying only one baby. About 20 women were carrying more than one babies. And we found that in this condition, if there is SARS-CoV-2 infection to a twin pregnancy, there's a very high chance of either delivering preterm or delivering there's a very significantly higher chance of delivering preterm. They were more likely to develop pregnancy-induced hypertension or preeclampsia or eclampsia-like conditions. So we report that if there is a twin pregnancy with SARS-CoV-2 infection, she is likely to have a more adverse outcome than her infected counterpart who is singleton delivery, and this group of women would require more special attention. This was something that Dr. Rahul and his colleague at NIRRH Hospital found that there was many women who were reporting with postpartum psychosis. So when women are pregnant and they deliver, once the delivery is completed, in small subset of pregnant women, there could be postpartum depression. But these were some women. So this is a case series of about 3, and there was one more here. I haven't shown the data, where they found that there was these women, because of SARS-CoV-2, they had a major anxiety and stress developed in them and developed postpartum psychosis. And about 35% of women with postpartum psychosis have posed their risk to infant. And in our case series that we published from our registry, we found that 2 women required separation from the infant, and one mother actually required a lot of supervision because she felt that her baby, she is going to kill her baby because she has SARS-CoV-2, and she was really getting depressed and psychosed, and this is a real problem. In that case, another one was feeling that everybody around her is trying to give her SARS-CoV-2, that she and her baby would die, and therefore she was undergoing a lot of psychosis, and she really did not like her baby, and there was a lot of problem in that case, and doctors had a tough time managing it. So just to tell, so in short, it tells us that there is a psychological involvement during pregnancy from SARS-CoV-2. We are collecting more data in this direction, that what is the prevalence of psychosis in this woman, but this is a problem because of SARS-CoV-2, we all have some sort of psychological anxiety, and when they superimpose with pregnancy, there could be more detrimental outcomes. Anyways, getting back to the main topic, our major interest here was we had this one case report where there was vertical transmission of SARS-CoV-2, that is from mother to child. The bigger question that we asked was that what is the overall incidence? Whether does it happen in all the mothers or it happens only a subset of mothers? We found, so we had data from our registry data, we had data of 807 neonates who were positive, who were tested positive, who were tested for SARS-CoV, whose mothers were positive for SARS-CoV-2, and these newborns were also tested for SARS-CoV-2 in their throat swab within first 24 hours of birth. And we found that about 6% of these babies are positive for SARS-CoV-2 when they're already born. So by the time that they're born, they're already infected with SARS-CoV-2. And this infection is coming from the mother. And very interestingly, about 50% of these cases, we could also found that the placenta and the umbilical stump was also positive. This means that it's not that all women would pass on the SARS-CoV-2 from the mother to the child, but in about 6% of cases, there is a definitive chance that SARS-CoV-2 infection is vertically transmitted. I must warn you here that this is data only from RT-PCR positive babies, remember, and also only tested in throat swab. Remember that we have now collecting more data with the help of our clinical collaborators in Bhatia Hospital, Dr. Kirti Kulkarni, where we have now collected maternal fetal diodes of a large number, and we are looking at multiple tissues, not just placenta, but even systemic viremia, whether the virus was there in the serum or not. And trust me, this number is going to be much, much, much higher. This is only positivity in the throat swab. Throat is not the most permissive site for SARS-CoV-2 in the newborn. The other permissive sites which have not been tested. So as of now, as of today, we claim that at least in 6% of pregnancies, near-term infected pregnancies, there is a vertical transmission of SARS-CoV-2. Now, the question that is asked is that, okay, these newborns are also infected with SARS-CoV-2. What is the general health of the newborn? And here is the data. We have data of 33 babies who were infected with SARS-CoV-2 and about 490 babies who did not have SARS-CoV-2 infection. Remember, all the mothers were positive for SARS-CoV-2 during pregnancy. And here you see that these babies are more likely to develop hypocalcemia and hypoglycemia at birth. The infected babies who have got the virus from the mother are significantly likely, and there's a 4 times chance that they would develop sepsis. The rate of NICU admission was not very different between the infected and the uninfected babies, but interestingly, we found that the SARS-CoV-2 infected babies spend much longer time in NICU as compared to their uninfected counterparts. And in NICU, they required more aggressive therapies like ventilation and CAP, which was not required at a very high rate in the uninfected neonates, and the risk of death. So if there is vertical transmission of SARS-CoV-2 from the mother to the child, the likelihood of the child death is almost 10% as compared to less than 2%, as compared to 2% in the uninfected counterparts, and this is statistically significant. So this tells us that when there is infection of SARS-CoV-2 in the mother, there is a likelihood that the virus is going to get shed into the circulation. From the circulation, the virus is going to get into the placenta. Into the placenta, the virus can happily multiply, mount an immune response, and it can also get transmitted from the mother to the child. And then when such children are born, they would be very likely to get admitted into NICU. They would require more aggressive therapies, and therefore the earlier belief that young babies are not infected with SARS-CoV-2 or they do very well is not the case. Our data suggests that young infants, newborns who have inherited SARS-CoV-2 perinatally are more likely to have adverse outcomes. Of course, we are collecting more data in this direction. This is very, very early data. A lot of this data is unpublished or under process of publication. But the take-home message from our studies are that the human placenta is permissive to coronavirus infection. In the placenta, SARS-CoV-2 can get infected. SARS-CoV-2 can infect the placenta, and it can replicate in there, and it can lead to mother-to-child transmission, leading to and also mounting an inflammation. The chance of vertical transmission of SARS-CoV-2 perinatally, that is near term. So remember this data, what I've told you, are women infected in the 3rd trimester. I've not segregated this data into 1st and 2nd trimester, but very, very early evidence suggests that the 1st trimester, there is more likelihood of adverse pregnancy outcomes. Hopefully in some point of time, we will have the data out from the registry. But the mother-to-child transmission risk during 3rd trimester infection is 6 to 7%. And in this, children with vertically acquired SARS-CoV-2 infection are more likely to have adverse outcomes, and these will need special attention. With this, I will stop here. I'm really grateful to the audience once more who has spent a really nice Sunday evening listening to my talk. But I'm also grateful to my laboratory staff, my students especially, who have spent all the time during lockdown working very hard within the lab or from their homes. My gratitude to Anshul and Nancy, who have contributed a lot to the placenta data. I'm also grateful to Dr. Rahul Kajbiye, who took the initiative of starting the pre-COVID registry, and it is through that registry that we have been able to get a lot of insights into the effects of SARS-CoV-2 during pregnancy. I'm grateful to Dr. Vainav Patel and Dr. Suchitra Surve, who have been very instrumental in giving us a lot of data on the RT-PCR front, and Dr. Prajakta Shinde. I've already told you about the data of the case report which she had given to us, and Dr. Mohit Jolly from ISC have helped us tremendously with the single-cell RNA-seq data. We are grateful to our director, Dr. Smita Mahale, who has retired, but she was the one who really pushed us during this lockdown to work and plan a lot of these things. And it is her vision that we have got these fruits. And I'm grateful, I'm eternally grateful to her for believing in us and letting us do a whole lot of work. And thanks to ICMR for continuously supporting us in all of our endeavors. And Anshul gets a lot of funding from his salary, and a lot of funding in the Placenta work is coming from DST, mainly the imprint sub-2 grant. And I'll be happy to take questions.
Thank you, Dr. Modi, for such an insightful, informative, and exciting. I'm running out of adjectives, to be honest, and a timely talk on how SARS-CoV-2 affect the developing embryos and its overall impact on pregnancy. And also the development of the pre-COVID registry that you and your lab has generated, which is immensely useful for the doctors and the clinicians to tackle this deadly pandemic. As I said, this is a special lecture on National Science Day, where we celebrate the scientific discovery of Raman effect. So on behalf of Biologically Speaking, let us all congratulate Dr. Modi and his team for doing an amazing body of work in such a short span of time during the pandemic. And what better day to celebrate this scientific achievement? And I'm not surprised to see the amazing questions that we have received in the YouTube chat box. And I'd like to cover most of them if Dr. Modi allows me and if time permits.
Sure, I'll be happy to answer them.
So our first question is from Ashwini Kumar. Is there any difference in effect of SARS-CoV-2 on the embryo in symptomatic and asymptomatic pregnant women? And there is a second question, what are the effects of this virus on endometrium?
Getting to the first question, are there any effects of SARS-CoV-2 on embryo in symptomatic and asymptomatic pregnant women? Ashwin, this is very difficult to get the answer, because at this point of time in early pregnancy, we can never get the embryos. All this data that's coming out is from IVF IVF embryos. It's just that as of today, we only have data where pseudoviruses have been incubated with the virus and infection is seen. But if the maternal status and early embryonic development, we cannot have any predictions here. But in the first trimester, so I showed you the case report where there is an asymptomatic woman and she's giving, and there is a transmission of the virus. Most likely that the symptomatic status of the mother is not a determinant of the pregnancy outcomes, is what we can say. But we are still analyzing the data. As of today, most data suggests that there is no relationship between maternal symptoms and pregnancy outcomes. I hope I have answered that question. Getting to the second question, what is the effect of SARS-CoV-2 virus on the endometrium? Unfortunately, there is just no data out there where people have looked at endometrial tissue and looked for SARS-CoV-2. But my collaborator and a visiting student in my lab, Patricia, she's now in Spain. She's published a paper where she's shown that ACE2 and TMPRSS2 are expressed by the endometrial cells, and there is a cyclic change during, and then there are changes in the menstrual cycle, suggesting that the necessary machinery probably is there in the endometrium, but whether the endometrium can mount, can get infected with the virus, there is again no data available there.
Okay, thank you. So the next question is from Joyan Raichaudhuri. Can the fetus have a long-term effect of COVID after birth if it got transmitted from mother during pregnancy?
This data is just accumulating. We don't have any long-term data of this, because hopefully from the registry, we should be able to have some information. But my last slide, I've already shown you that babies who have inherited SARS-CoV-2 from their mother, they're more likely to have adverse outcomes. They're more likely to be admitted to ICU. In the ICU, they're more likely to stay longer, and they may develop neonatal sepsis, and they require more aggressive treatment. But we definitely need a long-term data in this direction.
Okay, so the next is from Deepak Kumar Poria. What is the quantitative estimation of H2 positive TMPRSS2 positive expression of placental cells in relation to alveolar epithelial cells in the lung?
Yeah, so this was one question which we are persistently trying to answer when we were publishing the placenta paper. There's no straight answer to this because in the alveolar epithelium also there are various reports showing different huge variation in the number, but the average expression, the average numbers of pneumocytes in the lung is about 2 to 2.5%. In the placenta is about 20%. So it's far more in the placenta than the lung. But remember, the placenta at the same time is also protected because the lungs are directly exposed to the virus, whereas the placenta is not exposed to the virus directly. It has to get shedded into the maternal circulation. So the chance of it getting infected is lower, but once it sees the virus, the infectivity is going to be mounted. So that's how I would look at it.
Okay. Thanks, Dr. Modi. The next question is from Pat Puri. Apologies if I am pronouncing your name wrong. The IgG immunoglobulin can pass through placenta. Does it not stop SARS-CoV-2 infection in any capacity in the placenta and fetus?
A very, very pertinent question that now if the mother mounts an immune response, technically she should be able to clear it and the baby should not get affected. Even if the placenta is positive for SARS-CoV-2, because IgG can pass from the mother to the baby, the IgG should be able to clear off the virus in the fetus. Definitely yes, but as of today there are no direct correlates made. As we speak, experiments are ongoing where we are trying to correlate the fetus and the mother's IgG IgM status and the SARS-CoV-2 positivity status in the placenta. There is no data out there to say that how much of the protection is being acquired. We only know about the numbers of patients positive, but yes, it is going to mount a protective response. But remember, for that the mother has to first mount a response, and not all mothers during pregnancy mount an immune response. Plus the placenta does not like many immune things coming to it because it doesn't want to expose the baby to a lot of immune rejections. So there is no straight data again available. I'm sorry, most of my answers is there is no data because there's really no data there. But yes, it is likely, but soon we will have some information there.
Well, that's totally understandable. It's a very new topic that everyone started to investigating. Well, the next question is from Imroz Khan. Exposure to infectious disease at birth are known to contribute towards historical reduction in human lifespan before Industrial Revolution in Europe. So do we expect the same for COVID-2 infection at birth, something like accelerated aging, which is a very interesting topic?
Very, very interesting question. And I think that's the reason why we have started these registries. And a number of registries are started for this reason, because we need to be prepared for the pandemic. Today, our situation is this bad is because we did not pay much attention when the SARS or the MERS pandemic was there. Of course, we did not have these kinds of technologies. We did not have these many patients. So this pandemic has actually given us this opportunity. And hopefully from the registry data, we should be able to have this answer in time. Accelerated aging, because again, we really don't know. It's just that we only know that the mother can pass on the baby, mother can pass on the virus to the baby. And of course the placenta, we are also trying to now look at the placenta at the molecular level. We are trying to see what are the changes in gene expressions, and because the placental health can have long-term sequelae on baby's health, so it is likely that there could be something, but it is only time to come that we'll be able to get this answer.
Dr. Modi, if you still allow me to, there's a long list of questions.
I will love to answer all the questions.
Great, great. So the next question is from Debanjan Mukherjee. Considering lung development mostly takes place in the third trimester and postnatally, unlike alveolar fibrosis, what can be the reason for the fetal death?
So in the first trimester, the chance of fetal death, we believe, is because of the hydrops which has developed. So what happens is, now fetal hydrops is a very unique condition which develops because of a variety of reasons, including viral infection. And remember, at the first trimester, the placenta by itself is very leaky, and the placenta has mounted an immune response. So a lot of IgG, IgMs would have gone to the baby, and the baby would not like it, and therefore it is likely cause of an abortion. In the third trimester, yes, that could be one reason why the vertical transmission from mother to child, when we look at the throat swabs, appears to be much lower, just 6%, because as you rightly said, that the lung development is still occurring and the lung maturity is still not achieved at birth, and the number of ACE2 positive cells are believed to be very, very low in fetal lungs. So it is not because of the lung infection there is a chance of fetal death, but the fetus is going to mount a whole lot of adverse immunological challenges, and which is the cause of fetal death. And of course, placental damage and the damage to the spiral arteries is also going to contribute to it.
I think along the same lines, Satyat Pandey also has a similar question. Is SARS-CoV-2 is the only reason of death of the baby?
No. Maternal, perinatal deaths are because of a variety of reasons. One of them is SARS-CoV-2. In this case report, what we have presented, so I'm not clear with the question, but I presume if you're asking about the case report which we have presented, we have tried to rule out as many possible causes there. We have shown that it is no chromosomal abnormalities. We have shown that the common causes of hydrops fetalis are absent, so the mother is not infected with other known viruses which are known to cause hydrops fetalis. So we have ruled out many known causes. So it's just by ruling out we have narrowed down to SARS-CoV-2. But of course, I still stand with the idea that there could be some other reason, and it's a mere association. But it seems to be unlikely because there are others who are reporting similar findings as well.
So we have from Sana Jalili, can placenta be asymptomatic? I think which you have explained it during the talk as well. For example, when placenta is preserved for stem cells collection, can it would be a symptomatic carrier for future organs in case of SARS-CoV-2?
Okay, so the question that I'm trying to, if I'm understanding the question correctly, it is that when we possibly collect the placenta for stem cells, can these stem cells be infected with SARS-CoV-2, and then if we use the stem cells later on, could that be a reason of propagation? Remember, stem cells are not isolated from the placenta for any therapeutic reasons. Are usually isolated from the Wharton jelly, which is very rich in stem cells. As of today, we have no data to show that these cells can or cannot get infected with SARS-CoV-2. Experimentally, if we are talking, my gut feeling tells me that unlikely that these infected cells will survive in the lab dish, because the lab dish has a lot of other things, and it's only the proliferating cells survive, and these viral infected cells most likely are going to die. So it's very unlikely that it is going to be carried on further.
So we have a question from Sundaram Achari, who is also part of the biological speaking team, wondering how the dynamics of ACE2 receptor across all trimesters influence placental infection. And the second question, what is the cause behind severe placental viremia than that of maternal?
Okay. So this is one of the very pertinent questions, that whether ACE2 levels are same throughout pregnancy or they are changing. Our data suggests that the first trimester and the second trimester are nearly same levels of ACE2. By third trimester, the numbers of ACE2 positive cells are reducing, but they're not completely disappearing. There is a controversial report which says that there are no entry receptors for SARS-CoV-2 in the third trimester placenta, but that's no more true. There is now a lot of data, including that of immunofluorescence and in situ hybridizations, where people have shown first trimester infection. Remember, ACE2 is not the only receptor for the virus. There are multiple receptors, like basogen I've already told you, and NRP1 is another receptor which was identified in November 2020. And as we speak, again, my students are looking at NRP1 data in the human placenta, whether NRP1 is there. So although ACE2 might be declining, but if there are alternate receptors present, the likelihood of infection is fairly high. We did a systematic review in our lab where we tried to see what is the percentage of infected placenta in the first trimester, second trimester, and term. Our estimate is that it is around 10%, and these numbers don't change much. But remember that the data on first trimester is very low. I think it's only one of our case report and one more which is available. So it's about 10 to 15% chance that the placenta will get infected at term, and this is not probably relating to ACE2 levels, but it's most likely using an alternate mechanism. What could be the cause behind the severe placental viremia as that of the mother? So what we think is that the mother got infected. She was asymptomatic, but the virus somehow shedded into her circulation and reached the placenta. The placenta is acting as a decoy because remember the receptors are there. It is a necessary machinery for its replication and endocytosis and exocytosis. So the placenta is acting like a decoy, and the placenta is keeping the infection there, and the mother is clearing off in her throat swab much faster. And once a placenta is mounted, a lot of viremia because it is a necessary machinery, and then the adverse sequelae follows. I hope that has answered the question.
Yeah. I think the next question is from Pooja S. What could be the possible reason of retention of viral load in placental tissues even when maternal blood shows viral clearance, considering the rare event of hydrops fetalis?
Yeah, so the answer is again the same, that it is because the placenta, the first trimester placenta at least harbors all the machinery for viral replication, endocytosis, exocytosis, and all the genes which are interacting with the virus. And even we have now data to show that there are about 60 genes identified which are necessary for viral infectivity, and almost all the 60 genes are expressed in the first trimester placenta. So it's like kind of a very conducive site for viral infection, and therefore it is probably most likely developing more viremia.
Right. Okay. The next question is from Dr. Noble Kurian. Only 3% amniotic fluid is infected. We should concentrate more on how it occurs. It happened for a small percentage, but the placenta have every receptor environment for virus entry. But still, why that happened?
So Nobel, remember that when we talk about these numbers, 3%, 4%, 5%, these have largely come from very small studies of about 10 and 15 amniotic fluids tested. Again, the RT-PCR in fluids is very, very challenging. The RT-PCR technology that is used today is only for throat swabs. Not all the laboratories optimize it for the tissue that they needed. So these numbers which are estimated are low. Even when we talk about 3%, it is active viremia in the fluid. Remember, the virus doesn't like to stay very long in the fluid. As soon as it's going to see some cells, it's going to get inside there and it's going to act as a decoy. So getting virus in the fluids is less likely than that what you would see in the tissues.
Right, great.
But yes, we must concentrate on this multi-tissue tropism so as to understand mother-to-child transmission.
Okay, I think we're running out of time. We still have a lot of questions, and we do apologize that we're not able to cover all of them. I'll take two more questions and we'll end today's session. So the question is from Jatin Rawal. I think we are all now moving into the phase of vaccination. So will vaccination help to reduce infection chance in fetus?
A very, very interesting question. But do you know that women were excluded from all vaccine trials? In India, women are not vaccinated. There's a clear instruction from the government of India that pregnant women cannot be vaccinated for SARS-CoV-2. And that's all because several months ago, last year, people said that, oh, there is no adverse effects of SARS-CoV-2 in the pregnancy. And therefore, people never thought that pregnant women should be included in the trials. Because pregnant women were included in the trials, no trials have been done, so there is no data, and therefore we cannot vaccinate them. And this is one of the biggest blunders that we as medical fraternity and the scientific fraternity have done, and we must learn from this pandemic. So yes, vaccination hopefully will, in theory, it would. There are some data in the West where they've vaccinated women, and they've been, okay, the protection is shown, but mother-to-child transmission, we have, again, I don't know. I don't know a straight answer for that. But unless a woman are vaccinated, we'll never have this answer.
Well, I think that's the end of from the chat box. And as a moderator, I'll take this undue advantage to ask a question of my own. It's like, I may be wrong, but there could be a limitation in getting these human placental samples. Did you think of trying to model this COVID infection or any other viral infection using the 3D embryo-like models in vitro from embryonic stem cells?
Yes, so we are now getting into that phase. So with my collaborator, Dr. Abhijit Majumdar from IIT Bombay, we are developing a placenta on chip. We are also developing placental organoids in the laboratory, but we are still not set there. But definitely we are planning that we would have these model systems in the lab. By that time, hopefully we will have access to the virus, because we don't have a BSL-3 facility at the institute. We are building one. So hopefully by next year, we will have these systems ready and we'll be able to answer more fundamental questions in these regards. And of course, we are carrying out now single-cell RNA-seq and a bulk-seq analysis of the infected and uninfected placenta. And hopefully, it will, together, all this should be able to give us a complete story.
Well, that's great. Thank you. Thank you so much, Dr. Modi, for taking the time to answer all these questions and also for agreeing to speak in this platform. And I must say, we have nearly 100 people watching during the whole webinar, which is amazing to see, especially on a Sunday evening in India and also early morning in the United States. So big thanks to all for tuning into this inaugural lecture in this platform. We truly appreciate your support and participation. We have a conference coming up next week, March 6 to 7, on the eve of International Women's Day, a stellar lineup of women researchers. So please visit our website, www.speakingbiology.wordpress.com, to register for the conference. It is free, but registration is important to receive the details of the webinar. Also, do follow us on Twitter and Facebook, and also subscribe to our YouTube channel to stay updated with this channel. And I hope, Dr. Modi, you also enjoyed talking in this platform. Thank you very much for tuning in and for speaking in this webinar.
Thank you so much, Shona.
Thank you.
Bye-bye. Have a nice time ahead.
You too. Thank you very much.